Good afternoon. My name is Leah, like Professor Germain said, but my teammates call me Little. And today I'm going to be talking about my research on progesterone levels in blood versus the saliva of beef cattle. So before we begin, I want to just do a little bit of background information. What is progesterone? It's a hormone, and there's steroid hormones and non-steroid hormones. So this first picture right here shows a hormone that isn't a steroid hormone. So here's the hormone. And it can't go into the cell, so it has to basically talk to this receptor right here and send its message into the cell. But then a steroid hormone can go into the cell, so the other picture shows steroid hormones, and it just goes straight into the cell to deliver its message. This is important because hormones, they are the chemical messengers of the body, so they're responsible for most of the major bodily functions. And progesterone is a steroid hormone, so it just goes straight into the cell to deliver its message. Progesterone is produced by what is known as the corpus luteum. I won't go into that because it's a lesson for another time, but it prepares the uterus for pregnancy. It tells us a lot of information about the ovarian function in mammals, so that's why we test progesterone levels. In our research specifically, we used cows. So in cows, the estrous cycle is 21 days long, and most of those days are spent in the luteal phase under the influence of progesterone. The estrous cycle is just the reproductive cycle of the animal. So at the end of the cycle, if the animal is pregnant, then the progesterone levels, they will stay high. But if the animal is not pregnant, then they will decrease. This shows a picture of hormone levels during the estrous cycle. You can see that there's a solid line and a dotted line. The solid one is estrogen, but we're looking at the dotted one, which is progesterone. On the y-axis, you have hormone levels, and on the x-axis, you have the day of the cycle. So you can see that progesterone levels, they rise steadily throughout the cycle, and then they level off for a little bit, and then at the end they decrease, assuming that the animal is not pregnant. Estrous synchronization is basically just when you take all the females in a herd and try to get them on the same estrous cycle. And this is important so that you could set the date when calves are born, because you wouldn't want calves to be born in the dead of winter or something like that. And it's practiced in most livestock species. But as I said before, we use cows for ours. Problem is that there's issues in being able to synchronize all of the animals in the herd. So that brings us to a major question: how do steroid hormones travel? So when you test progesterone levels, typically you take a blood sample. And this is just when you take the blood sample, what it looks like. And you put it in a centrifuge, which is just a machine that spins it around really fast and separates it into 3 layers. So this is after you put it in the centrifuge. This bottom layer is red blood cells, and then there's very tiny layer right there, and that's white blood cells, and then at the top is the plasma. So then you take the plasma, and that's actually what you're testing for the progesterone levels, and you just kind of throw away the red blood cells and the white blood cells. But the problem is that progesterone might be sticking to the red blood cells, so if you throw those away, then you're throwing away some of the progesterone and artificially lowering the progesterone in your sample. So we started to do some research, and we found that there's a correlation between progesterone levels in blood plasma and in saliva. And this correlation is something that was found in both humans as well as livestock animals. So we want to figure out which one is a better sampling method. So accuracy is pretty important when you're deciding what you want your sampling method to be. So if blood is the most accurate at telling us progesterone levels, then we would want to use that. If saliva is the most accurate, then we would want to use that. But if they're both more accurate, then you would probably want to use saliva because it's more cost-effective, less time-consuming, causes a little bit less distress to the animals. So why does it matter? Well, like I mentioned earlier, there's been problems in synchronizing all of the animals. So we want to design an efficient synchronization protocol, and this requires a standardized collection method. So we want that method to be the most accurate and most efficient one. And also our research can tell us more about how progesterone travels throughout the body. So the objective of our research was to assess the relationship between progesterone levels in blood and in saliva. This first picture shows taking a blood sample, and the second shows taking saliva. And as you can see, the cows really enjoyed our time together. So in March and April of last year, we went and we took blood and saliva samples from 5 different cows every afternoon for 21 days straight. And before we took any of the samples, we put them in the head chute, which is this, and we put a halter on them, and that just ensured the safety of the cows and the researcher. And then we took blood from the jugular vein, and we used what's called a vacutainer, which is basically just a test tube with a needle. And we took saliva from around the teeth and under the tongue, and we used a specimen collection cup to do that, or a pipette if the animal didn't want to give a lot of saliva that day. So then we went back to the lab, and we put the saliva in the freezer at negative 20 degrees Celsius, which is negative 4 degrees Fahrenheit. And then, as I discussed earlier, we took the blood, we would centrifuge it, we collected the plasma, then we froze the plasma next to the saliva. In August, we came back to test our samples. And so we thawed all the samples, and we put saliva in the centrifuge so that it would remove the dirt from it. But also freezing and centrifuging saliva decreased the viscosity. So usually it's pretty thick and gooey, but by doing this, we made it thinner so that it would be easier to pipette. And then we tested each of the samples twice using an ELISA, which stands for enzyme-linked immunosorbent assay, but we'll just stick with ELISA. And then we ran some statistical tests. So we used a paired t-test to see if there was a large difference between saliva and blood when it came to progesterone. And we also did a Pearson correlation coefficient to see if they correlated. This is a little bit of how ELISA works. It seems pretty confusing, but I promise it's really not that confusing. So you have a plate that has all these different individual wells, and one sample goes in each well. And at the bottom of each plate is a plate-bound antibody, which is that gray Y over there. And then stuck to that is a progesterone-specific antibody, which is the green Y. So then we put our sample in the well, and the progesterone, which is the red dot in this, will stick to the progesterone-specific antibody. Then we add a tracer, which is the purple thing right there, and it sticks to progesterone. And then we wash all of that out, and we put a substrate in it, and the substrate interacts with the tracer, and it causes a color change. So the color change will tell us how much progesterone is in the sample, and we measure that color change using a spectrophotometer. To assess our method reliability, we calculated the coefficient of variability, or the CV, and there's 2 of these. The intra-CV measures duplicate consistency, and our goal was to get under 20%, but we got a little higher at 25.9%. And then the inter-CV measures plate-to-plate consistency. We wanted to get under 15%, and we got a little lower at 14.5. So these next few slides show graphs of the progesterone levels for each of the individual cows. On the y-axis, you have the progesterone levels in picograms per milliliter. Then on the x-axis, you have the day that the sample was taken. The blue line shows blood plasma, and the orange line shows saliva. And anywhere that there's a gap just reflects a sample that couldn't be read by the plate reader because its concentration was too high or too low. So as you can see from these first graphs, it doesn't really look like there's much of a correlation between these two, but you can see that progesterone levels in the plasma tend to be higher than those in the saliva. These next graphs show pretty much the same thing. It doesn't look like there's a correlation at all in these, but these graphs do a good job of showing the difference between the progesterone levels in the blood and in the saliva. You can see that they're a lot higher in the blood than they are in the saliva for these cows. And the last one shows the same thing. It didn't look like there was a correlation, but there does look like that there's a difference. So then we did our statistical analysis, and we found that saliva and plasma levels were significantly different. And we also found a Pearson correlation coefficient of 0.28, so that indicated there was not a correlation. A correlation coefficient that's 0.65 or higher would indicate a correlation, and anything close to 1 indicates a strong correlation. So since ours was so low, no correlation. The large difference between the progesterone levels in the blood and in the saliva was expected, but the lack of a correlation was unexpected because we read many articles that did find a correlation. One reason that we can think of for this is that we used an ELISA, whereas other studies used radioimmunoassay, or RIA. And they work by similar principles, except the ELISA uses the color change, and the RIA uses radioactive materials, so it's measuring gamma rays. A lot of times in older studies, they will use RIA because that used to be the most effective and it was the most accurate. But now ELISA is the most accurate at measuring really small amounts of hormones, and ours was in picograms per milliliter, which is really, really small. So we used ELISA because it's more accurate today. A limitation is that we did not know if many of the animals were cycling. I showed that graph earlier of progesterone levels during the estrous cycle, how they rise for a long time, then they level off and decrease, and none of our graphs really match that, so we concluded that most of our animals were probably in estrus. But this research does provide a foundation for future research, such as testing estrogen levels in blood and saliva to see if they travel in the same way as progesterone levels, and also testing an outside source of progesterone to see if that would travel the same way as naturally produced progesterone. I would like to thank the SCICU for funding my research, SWU, the Science Division, and the Honors Program, Clemson for allowing me to use their lab and their animals, and also Professor Stacy Johnson. Thank you.